Welcome to the Da Vinci Academy Histology video course. The entire video course is available on YouTube and covers all of the fundamental principles of histology and relevant cell biology. You can find all of the videos from the course by clicking the histology playlist link in the description below, and then you can access the corresponding practice questions and histology lab videos by going to our website, which is also linked in the description below. Alright, so this is the first of 4 lectures for the cardiovascular system. And in this first lecture, we're going to be talking about the histology and structure of arteries and capillaries. So first, just an overview of the cardiovascular system. It consists of the heart and the blood vessels and is responsible for providing oxygen and metabolic nutrients to the body. And then what it also does is, mainly through the venous system, is actually removes CO2 and metabolic waste products from tissues as well. So you have metabolism going on and you feed it through oxygenated and nutrient-rich blood. And then as a result of metabolism, you produce CO2 and other metabolic waste products that are, if they stick around too long, can be toxic for tissues. And so they get drained out by the venous system carrying blood back to the heart. So the flow of blood is obviously it's from the heart, and then you make it your way into the arteries. Then you go into the capillaries here, and then you go into the veins, and then you go back to the heart. Arteries always carry blood away from the heart, so artery A away, and then veins always carry blood to the heart. Usually arteries are carrying oxygenated blood and veins are carrying deoxygenated blood, but that's not always the case, as we'll talk about in a second here. The cardiovascular system consists of 2 dual circulations, and those are the systemic circulation and the pulmonary circulation. So the systemic circulation begins with the aorta coming off the left ventricle, and it branches off into all the different arteries that travel all throughout the body, perfusing all the different organs. And then those arteries eventually branch into capillaries. Capillaries is where oxygen is dumped off into the tissues, and then you have CO2 being produced by metabolism that gets picked up in the capillaries on the venous side and then gets brought back to the heart. Now the pulmonary circulation, this supplies blood to the lungs for gas exchange. So these systemic veins are carrying CO2 rich blood, and you got to get rid of CO2, so you're going to bring it up here to the heart, and then the heart is going to carry this deoxygenated blood, CO2 rich blood, through the pulmonary arteries. So here's an example where an artery is not carrying oxygenated blood. It's carrying very deoxygenated blood. So it's going to carry this here, and here in the pulmonary capillaries, you're going to release CO2 into the air within the lungs, and then the O2 that got breathed in will then enter in at the capillaries, and you'll oxygenate the blood again to then send it out to the rest of the body. So then the pulmonary veins carry very oxygen-rich blood back to the left side of the heart, and then that can enter into the systemic circulation, and the cycle goes on. So the general structure of a blood vessel, whether it's an artery or a vein, it has 3 layers histologically. So it has the tunica intima, which is the innermost layer to the lumen here. So here's the lumen. You have the tunica intima. Then you have the tunica media, which is the middle layer, the muscular layer. And then the tunica adventitia, which is the outermost layer, kind of the case or covering for the vessel. So if we look at it histologically here, this is a medium-sized artery. So you have the lumen here. And then you see this very thin layer here. This is the intima. Then you have the media here. And you can see that with the smooth muscle cells like this. And then this connective tissue layer here, this is the adventitia. So the tunica intima in general, its components, it's mainly composed of the endothelium, which is the layer of simple squamous epithelium connected by tight junctions. And you can see that here. So here's the intima layer here, here's the media here, and then this layer out here is the adventitia. So you can see comparison, especially in arteries, it's a very thin layer. If we zoom in here, so this is this portion here zoomed in. As you can see, here's the endothelium here. Here's the nuclei of the endothelium. It's very densely staining, ovoid shape. If you remember, endothelium is this kind of very thin, kind of wide-based cells like this. And there's only one layer of them, like this. And then they're selectively permeable, and then it often can be fenestrated. And we'll talk about that later on in the lecture. Below the endothelium, just like many other epithelial layers, there's a basal lamina to help secure it in place and provide support. And then below that, there's a subendothelial layer of loose connective tissue. So this portion in here kind of before you get to this border here of the media, this is all loose connective tissue in here, kind of loosely organized. And this contains the internal elastic lamina in arteries, which is a histological way to distinguish arteries from veins. And then the tunica intima is actually the site of atherosclerotic plaque formation, which we'll talk about in a little more detail in the clinical pearls section. So the tunica media, as I mentioned, is the smooth muscle layer, and it's arranged in concentric layers, meaning in circular layers like this, multiple layers around the lumen here, and then you have the tunica intima in here. And these are perpendicular to the vessel of the axis. So the axis of the vessel is coming into and out of the screen, versus these fibers are going around like this at a 90-degree angle. There's gap junctions between the smooth muscle cells to help facilitate contraction. And then you have sheets of reticular fibers, elastic fibers, and lamellae that are intertwined between the layers of smooth muscle cells. There's an external elastic lamina that surrounds the tunica media layer, and you could almost think of it as a border between the tunica media and the tunica adventitia. Smooth muscle regulates the diameter of the blood vessel, which then regulates blood flow. So if you go from here to here, and you have contraction of the smooth muscle, as you can see in the lumen here, the diameter of the lumen narrows, and so you're going to limit blood flow. So as you have a decrease in diameter, you have a decrease in blood flow. Versus if the muscles relax, so this would be called vasoconstriction, because the muscles are contracting and constricting the vessel. However, if the muscles relax, you have what's called vasodilation, and then the diameter increases. So you have an increase in the diameter, you're going to have an increase in blood flow. And this is regulated by the autonomic nervous system. And then just histologically here, again, here's that intima layer here. The adventitia is out here, and so in between here, this is your tunica media layer here. And you can see the smooth muscle cells here. Lastly, the tunica adventitia. So this is the outermost layer, and it's composed of dense, irregular connective tissue. You can see that down here in the slide. This is from a medium-sized vein. It's also composed of longitudinal collagen type 1 fibers with elastic fibers and fibroblasts. And it's the thickest layer of venules and veins, versus the tunica media is the thickest layer for arteries. That's kind of the key component or a key distinguishing factor between veins and arteries. For larger vessels, their tunica adventitia often contains a few additional features. One, the vasa vasorum, which are blood vessels that are actually running within the adventitia and provide perfusion for larger blood vessels, because these are very large diameter structures and they have a lot more tissue that needs perfusing. So you have this vasa vasorum, this kind of extra blood supply. And you find that in the aorta, superior vena cava, inferior vena cava. Then you can also have the nervi vasorum or vascularis, and these are unmyelinated sympathetic nerve fibers as well. So now we're going to talk about the functional and histological features of the different types of arteries and veins. So you start out with large arteries, which would be, for example, the aorta. And then you have, branching off of that, you have the medium arteries, which an example of that would be, say, the renal artery going to the kidney. Then, for example, the renal artery is going to enter into the kidney, and you're going to have smaller arteries. So you have small arteries that branch off from there to bring blood flow throughout the organ. Then off of the small arteries, you have the arterioles. And these are what bring blood to the actual capillary beds. So first you have large arteries. These are arteries that branch directly off the heart. So it would be the aorta and the pulmonary arteries. Histologically, what you have is some characteristics here of the thickness. So the tunica intima is actually pretty thick versus it's very thin in the other types of arteries. So you can see here's the lumen here. And then if you go from here all the way down to here, that's all tunica intima. And you can see it's very, very thick. And then it has a multi-layered internal elastic lamina. Now the tunica media is also very thick. So if you look here, this is intima, or we'll say here, somewhere in here, this is intima here. But then from here all the way down, even off the slide here, that's media. And so the tunica media in large arteries contains multiple sheets of elastic lamina. So elastic fibers organized in these sheets here. And so because of that, these are actually sometimes called elastic arteries. And what this elastic layer does, and you can see this here with these elastic fibers stained, you can see them stained in blue here all throughout this layer. So when you have a large bolus of blood being ejected directly out of the ventricles and into these large arteries, you need to be able to accommodate that. So you have these large vessels, they're going to fill up with blood. And as being elastic tissue, that helps recoil. So what's going to happen is it accommodates this large bolus of blood, and then it recoils back, and that actually gives you another push. It gives you that diastolic push, and it gives you that kind of diastolic pulse of blood. And what that does is that during diastole, that helps maintain continuous pulsatile blood flow. And then lastly here, the tunica adventitia is notable for containing vasovasorum in these arteries. Medium arteries, so these branch from the large arteries to provide blood flow to specific regions or organs. Examples would be the axillary artery to the proximal upper extremity, the radial artery to the more distal upper extremity, the femoral artery to the proximal lower extremity, carotids to the brain, renal to the kidney, mesenteric to the gut, coronary arteries to the heart. The tunica intima contains a prominent internal elastic lamina. This is actually a histological hallmark of these, because you're going to have to be able to tell medium arteries from large arteries from small arteries on a histology lab practical, so you need to know these key features. So in medium arteries, they're going to have a very prominent internal elastic lamina. We're going to show this on the next slide. The tunica media contains several layers, about 30 to 40 layers, depending on the vessel, of smooth muscle with elastic lamina that decrease in number as the artery size decreases. So the smaller the artery, the less layers of elastic lamina that are found. And so as you can see, here's the intima, very thin, and then here's the media here. And then you have an external elastic lamina that's not as prominent as this internal elastic lamina, but nevertheless, it provides a junction between the tunica media and the tunica adventitia. And you can see that here. So here's those concentric layers of smooth muscle, and then right here you have that dense irregular connective tissue found in the adventitia. Now what's important here is that the thickness of the adventitia can be about equal or maybe a little less thick than the media, and that's kind of characteristic of medium arteries. You can see that here. This layer is almost as thick as the media here. A couple other slides from a tunica media. This is actually from the same vessel. As you can see here, this kind of dark standing line just below the endothelium. This is that prominent internal elastic lamina. And then here, this kind of shows you more of all three layers. So again, here's that prominent internal elastic lamina traveling just below the endothelium. And then you have the tunica media here, which is this muscular layer here. And then as you can see here, here's that tunica adventitia layer here. Then you have the small arteries and arterioles. So small arteries, their tunica intima contain an internal elastic lamina versus arterioles do not. In small arteries, this isn't going to be as prominent as it is in medium arteries, so that's another way you can distinguish medium from small arteries. Small arteries contain up to 8 layers of smooth muscle, so not too many compared to the 3 to 40 in medium arteries. And then in comparison to arterioles, arterioles only contain about 1 to 2 layers, so much smaller. And remember, arterioles, that's kind of that last stop in the arterial tree before you get to capillaries. And then the tunica adventitia for both small arteries and arterioles is a very thin layer, and it actually blends in with the surrounding connective tissue. So here's the lumen here. You can see these red blood cells in here. Then you can see the intima layer, very thin media layer around. And then as you can see, the adventitia kind of blends into this surrounding connective tissue here. So the last branch in the tree of arteries is the arterioles, just before you get to the capillary beds. And the arterioles are actually very important in terms of the overall physiology of the cardiovascular system and regulating blood flow. And they actually are the key point of regulating blood flow through the capillaries. And they do this via contraction and relaxation of that smooth muscle layer in the tunica media. And so contraction is called vasoconstriction because essentially it's in the name. You constrict the vessel, you make the radius or diameter smaller. So this is vasoconstriction. And as you can imagine, this is to limit blood flow. And then you have vasodilation, which is relaxation of that tunica media, that smooth muscle layer. And that allows to increase blood flow. So increase blood flow, decrease resistance, decrease blood flow by increasing resistance. And what's important to know is this is actually the major site of vascular resistance, and they play a large role in both hypertension, which is high elevated blood pressure, and shock, which is at the simplest form, decreased perfusion to your organs. And to illustrate how this changing the radius of the blood vessel can have such a major impact on resistance and why arterioles are such major sites of vascular resistance, it's important to know this equation. You don't have to memorize it, but understanding the principles of it is important. Resistance is proportional to, this is a Greek symbol for that represents resistance. This is length of the blood vessel, and then obviously R to the fourth power, radius to the fourth power. So if you increase the length of the vessel by 2, you'll increase the resistance by 2. However, if you were to change the radius from 10 to 5, what you're doing is changing from 10 to the fourth, which is 10,000, to 1 over 5 to the fourth, which is 625. And so you're going from 1 over 10,000 to 1 over 625. As you can see, that's a dramatic difference. And so, again, this is what resistance is proportional to. So by decreasing the vessel, so again, by decreasing the radius, you're going to dramatically, as a result of this radius to the fourth power, you're going to dramatically increase the resistance. You can see why even making this small change in the radius dramatically increases the resistance. And as you can see, then plays a larger role in terms of the overall physiology of regulating blood flow through capillaries. Another thing we mention here briefly to know is met arterioles. These are short vessels that link arterioles to capillary beds with thickened tunica media called precapillary sphincters. And these are essentially to regulate capillary blood flow, but also to shunt blood flow around capillary beds. So actually, if we draw in a little diagram like this, so this is an arteriole here, and we'll draw a met arteriole here, and then this will give rise to capillary beds here. And so out here, this is a capillary bed here. And so the capillary bed will obviously end up in a venule. So in blue here, come out here, and this is your venule here. Now this portion here is the met arteriole. And what they have is actually these, as we mentioned, these precapillary sphincters, which I'll draw in with these lines here. These can constrict and limit blood flow to the capillary bed and essentially shunt it directly to the venule. And so this helps with shunting blood flow away from capillary beds to other areas where it's more needed. So capillaries themselves, they're the smallest diameter blood vessels, and they're the site of exchange of gases, metabolites, and waste products between the blood and peripheral tissues. So if we draw a capillary bed down here, and then you obviously have the blood going through here, and then you have tissues here. So obviously you have metabolism going on, right, to power the cell, generate ATP for energy. And so for metabolism, you have aerobic respiration, right, that needs oxygen. So you will drop oxygen off from the blood here being carried by hemoglobin. So you'll exchange O2 there, and then metabolism generates CO2, and so that'll get picked up in the capillary beds. The other thing metabolism generates are waste products. And so those will get cleaned up and carried out by the blood. And so the CO2 and the waste products get carried in the venous blood. The CO2 gets carried to the lungs where you breathe it off into the atmosphere. And then obviously the waste products, they get carried to either the GI tract or the kidney where they can be excreted. Histologically, they consist of a single endothelial cell. So if you recall, you have your endothelial cell like this. You have your nucleus in here, and it's a single cell. And you can see, and sometimes this is just small enough that only one red blood cell can pass through. That's why red blood cells need to be so flexible, because sometimes they need to pass through these very narrow capillary vessels. The density of capillaries within tissues is dependent on the metabolic demand of the tissues. So since this is the site of where you exchange oxygen for CO2 and pick up waste products, and then also provide nutrients. So you can also provide, obviously you eat a meal, you have glucose, you need to provide that to tissues, so you can drop glucose off into the tissues here at the capillary beds as well. What's important is to remember, such as tissues like cardiac muscle, obviously the heart has to beat all the time and every second of someone's life. So very high metabolic demand. As a result of that very high metabolic demand, heart tissue has very high capillary density. So a lot of capillaries present because you need to be efficiently bringing oxygen and glucose and other nutrients into that cardiac muscle, and then also clearing out the CO2 and the waste products. Capillaries, they're classified into 3 categories based on their permeability. This is the least permeable to the most permeable. So you have continuous, fenestrated, and discontinuous. And we'll go through each of these on the subsequent slides. So the first of these classes of capillaries are the continuous capillaries. And remember, these are the least permeable. And the endothelium in these types of capillaries are bound together by occluding or tight junctions, and you can see those down here in the diagram. You see these black lines between the different endothelial cells. These are tightly adhering each cell to each other, and it's limiting the exchange of materials between the blood and the tissue, and you'll see why that's important in a second. The other thing is that this endothelium is surrounded by a continuous basal lamina to also help limit that exchange. You do, however, have pinocytic vesicles, which are part of a process called pinocytosis, which is essentially quote-unquote cellular drinking. These vesicles are found beneath the lamina and the basal membrane surfaces to facilitate the exchange of larger molecules between the blood and the surrounding tissue. So if you have a cell like this, and let's say you have a material like this triangle, and you want to get it from the blood here to the tissue. So what will happen is that this will come in close proximity to the cell, and the cell will start forming this vesicle that'll essentially engulf this material. And then what happens is it forms a vesicle within the cell that's carrying this molecule, and then the vesicle goes to the other side and fuses with the membrane, and then allows this molecule to be released to the other side, essentially transporting it from the blood to the tissue. So continuous capillaries, they're found in the central nervous system. What's important to know is they form the blood-brain barrier. The blood-brain barrier helps tightly regulate which molecules enter and leave brain tissue. And that's important because some molecules, for example, like glutamine, can be used to form glutamate, which is a neurotransmitter. And so you don't want to just have neurotransmitters or molecules that can be used to form neurotransmitters freely coming across the blood-brain barrier from the bloodstream into the brain tissue, because then you would be triggering signals in neurons that you maybe didn't intend to. And so you want to keep things tightly regulated in the brain. The brain's obviously a very sacred organ, very important organ, and you don't want to have unintentional signals being started that could cause dysregulation of certain processes. Continuous capillaries are also found in the lungs. They're also found in muscle tissue and the cortex of the thymus. We'll go into that in the immune lectures. Fenestrated capillaries, it's in the name. This endothelium has numerous fenestrations, or these little holes that allow passage of molecules. Again, you want to be thinking about these classes of capillaries, which organs they're found in. These are going to be found in organs where it's really important to facilitate rapid exchange of molecules. They are still surrounded by a continuous basal lamina, and they're found in the kidney, where obviously you're doing a lot of reabsorption. The endocrine tissue, where you're creating hormones that you want to secrete into the bloodstream efficiently. So it makes sense here. It allows you to efficiently secrete those hormones. The intestinal villi, where you're obviously doing a lot of absorption, just like you are in the kidney, so it makes sense. And then the choroid plexus as well, where it's important to facilitate rapid exchange of molecules. Discontinuous capillaries, these are endothelium with large fenestrations. You can see this here. And then no diaphragms. They also have these discontinuous gaps that exist between the endothelial cells, and you can see that here. These are called interendothelial gaps. And again, these are found in places where it's important to really allow the blood to just freely exchange with the tissue within the organ. The basal lamina itself is often discontinuous and sometimes even absent. You do have reticular fibers present. What this does is it enables easy phagocytosis of blood molecules. And so you can have cells here, such as macrophages. In the liver, these are called Kupffer cells, and they can phagocytose certain molecules that are coming in and out, including pathogens that could cause infection. These are found in the liver, like I've mentioned. Obviously, in the liver, you're essentially filtering the blood for toxins, and then you need to freely exchange certain molecules that need to be metabolized or processed within the liver or filtered out in the liver. The bone marrow, obviously you're synthesizing a lot of different cells, red blood cells, white blood cells, platelets, and so you want those to easily be able to enter the bloodstream. And then the spleen as well, you're doing a lot of filtering of the blood on a lot of immune processes occurring. Pericytes, we'll spend a little bit of time. This isn't super high yield, but these are a mesenchymal stem cell. That's one of the main facts you want to remember. They surround capillaries, and they're capable of differentiating into endothelial or vascular smooth muscle cells. They can be used to regenerate damaged endothelial cells or vascular smooth muscle cells. They tightly adhere to the capillary endothelium via numerous cytoplasmic processes. If you looked at these on electron microscopy, you'd see a lot of these different processes, finger-like processes like this from the cytoplasm. And then what's important to know is they're enclosed by the capillary basal lamina. So they're between the cell and the basal lamina. And so if you look at the capillary like this, here's the endothelium. The pericyte would be found in here, and then you have the basal lamina outside like this. Thank you for watching this video from the Da Vinci Academy Histology video course, which is completely available on YouTube. To access the corresponding practice questions and histology lab videos, go to our website using the link in the description below.